Hi everyone, I'm going to talk about the motility of bacteria lab. So in this lab, you're going to examine if bacteria are motile or not. And basically you're going to look at them under a microscope using a technique called hanging drop technique, which is shown in this image, and I will explain it. And with this technique, we're able to look at bacteria under the microscope and we can determine if they're motile or not. Remember that bacteria are motile if they have flagella. Flagella is just one type of structure that gives bacteria motility. And here are some images of bacteria with flagella. So we'll talk about them and we will start off with some background. To start off, not all bacteria are motile. Some are motile, some are not. Obviously, having motility for bacteria is an advantage because it can help you swim towards good environments, such as environments where there's nutrients, there's good oxygen, and swim away from harmful things. So flagella are these little structures that are stuck in bacteria, and they're stuck to the plasma membrane of bacteria by a basal body. I will go over their structure in this image. In these images here, we differentiated gram-negative bacteria from gram-positive bacteria. I know this is gram-negative bacteria because the cell wall shows an outer membrane and only gram-negative bacteria have an outer membrane. This is gram-positive bacteria because I just see the cell membrane, and outside the cell membrane, I see the peptidoglycan. Regardless of either, we see that in the plasma membrane, in the plasma membrane, anchored to it is this structure called the basal body. So we have the cell membrane, we have the basal body, which is anchored in the plasma membrane. So here's the plasma membrane of gram-negative bacteria. Here's the plasma membrane of gram-positive bacteria. Then we see the hook, and then we see the flagella. So in either, we have, again, the basal body that's in the cell membrane, then we have the hook, and then we have the flagella. So this is all the flagella components. The flagella is made up of a protein called flagellin, and this protein is what helps bacteria move. So bacteria can have different flagella arrangements. They can have either just one little flagella, or they can have multiple flagella. They can have hundreds of flagella. The flagella also can either be arranged polar. Polar means at an end. So you can have flagella at one end. You can have flagella at two ends. That's what polar means. Or the flagella can be arranged all around the cell, and we call that paratrichous. So for example, when we look at the bacteria E. coli, we see flagella all around. So that's called paratrichous flagella arrangement. And we see flagella in all types of bacteria. We can see them in bacteria that are rods, bacteria that are spiral, bacteria that are spirochetes. I just want to mention that in coccus-shaped bacteria, which again are the round-shaped bacteria, flagella are not that common. So coccus bacteria can still be motile, but it's not that common. So most coccus-shaped bacteria or round-shaped bacteria are non-motile. Okay, now to get into the technique in lab of how we actually determine if bacteria are motile or not. So to determine if bacteria are motile or not, there's a technique in lab called hanging drop technique. And it's called hanging drop because you literally hang a drop of culture from a slide. So you have your bacterial slide and you have your drop of bacterial culture hanging on it, and you look at it under the microscope. So that's what the technique is called. And with this technique, you suspend bacteria in a drop of fluid. It's a drop of culture that the bacteria is growing in. And in this drop, this is beneficial because you reduce evaporation and you reduce water currents so that you get the bacteria suspended in that drop of water. That's why it's much better than a wet mount. If you remember, with a wet mount, you basically just smash the drop of fluid that your bacteria is in. And with this technique, there's no fixing, there's no staining involved, meaning that we have the bacteria alive so that we can examine motility. Because remember that if we stain them or we fix them, you're killing the bacteria. So by killing them, you lose the ability to see if they're motile or not. And just to keep in mind, rod-shaped bacteria generally swim forward, and coccus-shaped bacteria, if they are motile, they do this spinning. It's harder to tell if coccus are motile. Rods are very easy. They just swim forward. And with this technique, we will be able to see if bacteria are motile or not. So I'll explain how the technique is done in a second in a better slide. This shows the hanging drop technique. And I want everyone to keep in mind that when we do this technique and we look at it in lab, you don't have to see all the bacteria be motile to determine that that bacterium is motile. Even just one bacteria moving means that the whole culture is motile, because a lot of times the culture will stop moving, they'll start dying over time. So that's why even if you see just one or two out of thousands of bacteria motile, it means that the whole culture is motile. So this is the hanging drop technique. And basically what you do is you have this slide. The slide is called a depression slide. So in the hanging drop technique, we use a depression slide. And it's called a depression slide because when you look at this slide, there's a little depression or opening. So that's the slide that you use. The way you do it is you take a cover slip, and on your cover slip, you put a drop of your culture, and on all four corners, you put a drop of water. Then you take your depression slide and you gently tap it over your cover slip, and you flip it, and you're ready to look at it under the microscope. I want to take note that when we go look at it under the microscope, and I'll state how that's done, we want to make sure to not confuse movement, motility, bacterial motility, with what we call Brownian motion. Brownian motion is just useless movement that we see where drops of water are just colliding with each other. So you think you're seeing something moving, but in reality, it's nothing moving. It's the equivalent of seeing a speck of dust in a cup of water and moving. The speck of dust is dead, it's not motile, but it looks like it's moving. So when we look at the hanging drop, the depression slide under the microscope, we want to make sure that we're really taking a good look at the bacteria, making sure that they actually are moving. The bacteria that you would examine had you been doing this in lab is we wanted you to determine if these groups of bacteria, if they're motile or not. So for example, you're going to look at Bacillus subtilis, Enterobacter aerogenes, Klebsiella pneumoniae, Micrococcus luteus, all of these bacterial species, and determine are they motile, are they not, using hanging drop technique, which again uses a depression slide, and then determining if they're rod or coccus, because you can tell if they are rod or coccus shaped from doing this technique. Now, obviously, if you were in lab, everyone can Google this on their phone, but the idea is that you learn how to do hanging drop technique. So the way this is done is, I have a video demo that shows how it's done, but basically you start off, once you have your hanging drop slide, to look at it under the microscope, you start at the lowest magnification, which is 4x or 40x total magnification, and you center your drop of water. You make sure that the iris diaphragm lever is all the way down. You want to make sure that you lower the light all the way so that you are able to see something, because the drop of fluid is transparent and your slide is transparent. So if the iris diaphragm is up, it's very hard to see. And then you slowly build up your magnification to 10x or 100x total magnification, and you center one edge of your drop of bacteria. And then the highest magnification you're going to go to is 400x total magnification, which is 40x magnification. And then you're going to focus on one edge of the drop and you're going to look at it. So these images are not very clear, but for example, in this image, if you really look close, there are rod-shaped bacteria. And these are still images, so obviously you cannot see motility with them. And I will show you a video. So we try to focus on one edge in the microscope. And here I have the little rod bacteria, they're here. We try to look at one edge, not the middle, because on one edge you can see a 2D image so that you're not looking at a 3D image of bacteria. And it's much easier to see motility with a 2D image. So here's a picture of rod-shaped bacteria. Here are coccus, very circular bacteria. And here's a video I took of a slide I did. So my hand was shaking a little bit, but you can tell. So these are rod-shaped bacteria, and if you really look closely, you'll see some of the bugs or bacteria shooting across. And with motility of bacteria, again, just one or two bacteria shooting across means that the whole culture is motile. Okay, so the objectives of this lab, what I want everyone to get is know if organisms are motile or not under the microscope. Now for the online class, you're not using the microscope, but I want you to have an idea from the bacteria are showed which are motile and which are not. So I want you to look them up. I think it's important to know that. And then the name of the motility procedure that scientists use in lab is called hanging drop technique. Hanging drop technique uses a depression slide. Know which bacteria were motile or not. So I want everyone to look up which bacteria are motile and which are not from that list. For hanging drop, I have a question. Is it better to do wet mount or for determining motility, I want everyone to know, is it better to do hanging drop technique or wet mount? Why or why not? So I discussed that. And in the hanging drop technique, I want everyone to know that we're only able to see motility. We're not actually looking at flagella. Flagella are very tiny structures that you cannot see. You can only see them if, for example, you do a flagella stain, which is Leibson's technique that we learned about earlier. If you're checking if unknown bacteria are motile, I want everyone to answer, should you fix them? Should you stain them? Why or why not? So just make sure you guys know the answers to all of these and you take time looking at the demo video of myself showing you how it's actually done.